We'll see. We'll see how it goes. All right. So welcome finally to Biochem 2, and buckle up because it's going to be a bumpy ride this semester. This is probably the second most challenging course on campus. Why? Not because any individual component of it is especially hard. Like I look at PChem when you got to do a lot of calculus in there. PChem 2, that is a very complex stuff here. Fortunately, this is not super complex to figure out any individual component. The issue is we're going to synthesize a whole bunch of different pathways together, and it's very easy to get them a little bit mixed up. So we're going to try and start off not too bad, but it does build, and it builds, and it doesn't build out, it just builds around in a big circle, like a big globular protein. Ooh, I'm going to reference Biochem 1, right? So this first chapter about bioenergetics is kind of the, so we're going to do this one. Here we go. Yep. Is almost like a review of Biochem 1. But it's going to emphasize our key principle here that if we look at the thermodynamics, and you guys will probably be happy because we're not going to look at very much thermodynamics, but if we look at the thermodynamics of the pathways we're talking about, we're going to see why they function the way they do and how we can utilize the thermodynamics to regulate them, along with some other stuff as well too. We got our common organic principles. We're gonna, again, define our high energy molecules, what they do there, and how we store energy, okay? So, come on, maybe, there we go. All right, so you might recall this figure was probably like one of the first ones from chapter 1. If we have a living organism, it needs a source of energy, okay? And ultimately, everything on Earth, it gets its energy from the sun. We can either have things that are directly photosynthetic, like our plants, which are kind of sleeping now, or our heterotrophs, which will eat the plants or otherwise get energy from someplace else, okay? So if we look at metabolism, which we're going to do for the first 3/4 of this course, we have a couple things that go on. We have anabolism, where we build up more and more complex molecules, and catabolism, where we take complex molecules and break them down. And ideally, we can couple these so that the breakdown will release energy, which can be used to drive the buildup. Because keep in mind, entropy, we always want to go towards more disorder. So when we actually make more complex things, we're going against entropy. But as long as we are, harnessing that energy from the breakdown of other stuff, our entropy part of our equation gets compensated for. I'm having a rough week too, so my brain is not necessarily functioning yet today, but we'll get there. I hope we will. All right. But again, catabolism, we have our energy depleted products. We kind of break things down and suck the energy out of them. And our anabolism, we build them up to make something that's more complex, and that is functional for living organisms. And we'll see our connecting molecules in here, like adenosine triphosphate or NADH and stuff like that, is where we get our energy from. And these are examples of high energy molecules. They can be broken down to provide energy to couple the anabolic reactions to build things up. All right. There we go, click there, okay. All right, so living organisms cannot create energy. They cannot destroy energy, just like normal thermodynamics. We can't create or destroy it. We can only transform it from one form into another. So transforming that, we have to increase the entropy, kind of like the stuff I just said here. So we're not gonna sweat that too much. Yeah, it's funny. I always like to keep this little cartoon in here because, when my son was born, my wife was in pharmacy school. So she had him sit on her lap and read about, various drugs. And that's how his pronunciation came about. Anyway, you ever get kids who ask, why is the sky blue? You know how to answer that, right? Rayleigh scattering. So yeah, there's a joke about that, but I've actually done that. So anyway, so just in the same way that the people who make school decisions about, hey, there's still wet roads, it could freeze overnight when it's going to be 40 degrees overnight. Maybe we need some more science people in the school system to say, no, it's not going to freeze when it's at 40 degrees. Anyway, and it rhymes. Anyway, if we go back to our equilibrium constant, which we're not going to go into much detail here, the big key here is if we look at our free energy, that's going to determine whether a reaction occurs spontaneously or does not occur spontaneously. So if this delta G is negative, we have a reaction that's going to occur spontaneously. It's going to go from reactants to products. On the other hand, if it's positive, then it's not going to occur spontaneously. And how do we get those reactions to occur? We can couple them with something that's going to provide the energy to get over that energy barrier, to force them, to get them to occur. All right, so this is probably I need to go trim out all the unnecessary slides in here, but this is the big takeaway right here. If our delta G is negative, we're going to go forward. If it's positive, we're going to go in reverse. And if it's zero, it's at equilibrium. We will almost never see a delta G at zero in a living organism. Now, that's not completely true because I have a couple examples of those in here in pathways, but then we start getting, our delta G not double dagger, where we look at the environmental effects and all that good stuff in there to see whether it's going one way or the other, okay? Usually, our reactions are far from equilibrium, okay? Here's just some examples of them. If you look at things like, acetic anhydride plus water to give us acetate, that is very favorable. It's going to just zip on through there. Same way, if we start burning palmitate, we get tons of energy out of there, which again kind of shows why we store energy as fat, because you can get so much energy from fat from oxidizing it. All right. And again, we measure these typically in kilocalories per mole or sometimes joules per mole, kilojoules per mole. Hydrolysis reactions tend to be very favorable, so they will often occur spontaneously. Isomerization reactions, where we're kind of rearranging the molecule, tend to have smaller free energy changes. A lot of time, I know we talked in Biochem 1 about our glucose and how it can be linear and it can circularize and then go back to linear. That is a very small free energy, so it's kind of constantly opening that ring and closing that ring to make our linear versus circular form. And of course, complete oxidation is strongly favorable. This is how most of we chemotropes get our energy. We essentially oxidize stuff with oxygen, and in a living organism, we want to make sure this is stepwise and controlled. You guys ever hear of spontaneous combustion, where someone just spontaneously bursts into flames and is reduced to a pile of ashes? I always thought that's where the biochemistry just goes completely wrong, and we have uncontrolled, massive, oxidation that happens there too. So theoretically, that is possible, but statistically, no, it's not possible. It would require, an infinitesimally small percentage of that happening because of all the regulation that goes on in here. But it's fun to think about. One day you'll just go in the lab and there'll be a little pile of ash there and go, oh no, where's Dr. Ebert? Okay. Okay. Oh, and again, there is an important point down here at the bottom. Being thermodynamically favorable is not the same thing as being fast. Some reactions might be very favorable, but if they have a high activation energy, they're going to proceed very slowly, which is why, again, we can use our enzymes to help facilitate the speed of that reaction. Okay. Energetics within the cell are not standard. So all those little delta G dot, delta H dot, all that stuff in there, we are non-standard. The actual free energy depends on the standard change as well as actual concentrations in there. So we can add this extra term in here. Our delta G is really delta G dagger dot here, where we include a term for temperature and our concentrations of our various reactants and products in here. Okay. If we have a pathway, which we will have lots and lots of pathways this semester, our standard free energy changes are additive. So if we want to look at the pathway of A to C, and A goes to B as an intermediate, and then B gets converted to C, we can look at the delta Gs for each individual reaction in there. So when we talk about glycolysis, we have a 10-step reaction, all right? And we can add up the delta Gs for all 10 of those steps to give us our overall energy. There will come a time where I have a question for you guys, like, well, how much energy are you going to get out of this reaction, out of this pathway? And I just kind of want to see how you do that. There are actually a couple different answers because it depends on which particular part of the pathway is in there. But when we get to glycolysis, we'll talk about the investment phase where we actually have to put energy in, and then the payoff phase where we start getting more energy out. So it's kind of like the saying, you got to spend money to make money. Well, for glycolysis, you got to spend a little energy to get out more energy in there. All right. We look at chemical reactivity. Most of our reactions, we're going to talk about similar things here where we have like the cleavage or formation of carbon-carbon bonds, cleavage and formation of polar bonds. We might see like our hydroxyl groups in there, or phosphate groups, or addition or transfer of groups, internal rearrangements, and of course our redox reactions. All right, so the good news is we don't have to cover every single possible reaction we could have. Living organisms, we look at a few and then just how we regulate them. All right. We should also define some terms. Homolytic cleavage, if we have a bond, we split it evenly. You get an electron, you get an electron. This is very rare. Normally, we don't want to split electron pairs like that because we end up making radicals, and radicals are highly reactive, so they can cause damage. So normally we'll see heterolytic cleavage where an electron pair goes one way or the other. So we might see a carbanion formation and the release of a proton, or we might see a carbocation and the formation of a hydride. Right? Where we can have something like this if it's a carbon-carbon bond, a carbanion, and a carbocation formation. Okay. I always comment, I had an advanced organic class and it essentially became follow the electrons. If you could figure out where the electrons were going, you could figure out what the products were going to be. Right? So that kind of holds true in biochem as well too. If we look at nucleophiles, they tend to be negative or partially negative, so they are attracted towards positive charges, and electrophiles tend to be positive or partially positive, so they tend to be attracted to negative charges. So you can see here our oxygen with a lone pair on it has a negative charge. It's going to want to interact with a positive charge. Anything that has a lone pair is typically going to be attracted to that. If we look here we have a carbon that's got 4 bonds. It's like, well, it's balanced, right? Except oxygen is more electronegative. So it's going to be electron withdrawing. And it's going to share those electrons with carbon, but it's not going to share them evenly. It's like a big kid and a little kid sharing toys. The big kid is going to have those toys more often. So the oxygen has a partially negative characteristic, meaning that the carbon in this case is partially positive. So it's a good target for a lone pair to come and interact with and maybe kick off one of those other bonds in there. Yay, organic chemistry, right? Yeah, okay. And we can see that, and we'll see that in here, like, for example, our phosphorus, when we talk about the hydrolysis of ATP, that's what's going to happen. We're going to have a lone pair that attacks the phosphate in the phosphorus in the phosphate group, and then kick off one of those oxygens, which kicks off a side chain, either 1 or 2 phosphates, phosphate or pyrophosphate. And again, yeah, back to Latin, right? So again, back in the old days, you needed to study Latin and Greek to go to college, and I'm old enough that in order to get a chemistry degree, we had to have a year of German because so many journal articles were published in German, and we never read any in German because they were all translated at that point. So they dropped that requirement either while I was there or shortly after I graduated. But it's still fun to try and go read journal articles in the basement of the library that were written in German. They were also very short, so that made it fun. All right, and then here are just some examples of them. Our classic aldol constant condensation, our Claisen ester condensation, our decarboxylation. I include these not because I want you to specifically know what these are, because you guys know what they are, yeah. But just as examples of how we follow our reaction here. So in this case, we have our carbanion with the lone pair there that is going to be attracted towards this partially positive carbocation. So we're going to see that carbon-carbon bond form, and we're going to kick off one of these bonds here so that we add a proton so that this carbonyl becomes a hydroxyl group. Okay. Okay. Same way we can see here, we can kind of follow these electrons, and we can see maybe here our lone pair jumps up this way. get other stuff in there. So just make sure that you're comfortable kind of looking at the way these reactions proceed. Okay, we won't go into excruciating detail about these, but there will be a few places where it'll be helpful. Okay, if we look at addition and elimination reactions, addition, we're adding something, eliminating, we're taking something away. So it just kind of looks at that, sp3 carbons, sp2 carbons, all that good stuff in there. And we can have SN1 or SN2 mechanisms for these reactions in here. So for example, if we look at isomerization of glucose 6-phosphate, because at one point we're going to convert glucose 6-phosphate to fructose 6-phosphate, we are essentially moving the carbonyl group from carbon 1 to carbon 2, okay? And we can't just go pull it off of there. We actually have a more convoluted process in here where here we have, some acting base, in this case, our enzyme, base 1 and base 2, will provide these lone pairs, which can help stabilize, or in this case, a hydrogen that will help stabilize this interaction and move this stuff around, and then we can add it back at a different position. Okay, so the net effect is we are essentially moving this carbonyl from carbon 1 to carbon 2, but first we have to pull it off of carbon 1, regenerate carbon 1. We actually make a carbon double bond between carbon 1 and carbon 2, and then we add our oxygen back across this double bond to get our carbonyl group in there. Leave that first one as a hydroxyl group. Feel free to interrupt me at any point because I know I'm jabbering away here. Group transfer reactions can be as simple as a proton transfer to as complex as a myristal group or something crazy like that. We'll often look at methyl groups, acyl groups like glycosol and phosphate groups in there. We'll see these as being important in signal transduction as well too. It's kind of like lightning and signal fire, turning on a light. We had a phosphate group. It's like, oh, turn it on. Take away a phosphate group, we turn it back off. Okay. Sometimes we'll see this pentacovalent intermediate where we have 5 bonds in there. It's like, what? It's like only 4 beats in a measure. Okay. We look at how we phosphorylate something. Here we have ATP. Well, our end phosphate in that ATP makes a good leaving group. Actually, we can actually have a pyrophosphate in there as well too. But we can donate that to phosphorylate something. If we hydrolyze ATP, it's very favorable under standard conditions. So we can have water attack this phosphate and clip off phosphate bond, a phosphate group in there, and leave behind, ADP or TDP or CDP or GDP as well too. We use adenosine because it's the highest concentration of the cells in there. If we look at this releases a lot of energy. Our delta G down here is about 30 kilojoules per mole. So that is what we call a high energy compound in here. It's not the highest, but that's another reason why we see ATP coupled with a lot of reactions, because 30 kilojoules per mole is a good amount of energy. And we can couple that hydrolysis with energy requiring unfavorable reactions to kind of drive them forward. Okay. We don't talk about this too much, but I just want to reiterate that magnesium is important for our triphosphates because the magnesium cation helps balance those negative charges that are stabilized on the phosphate groups in there. So again, our actual delta G differs from our delta G naught in there. So we're going to have a whole bunch of stuff that factors into this. And we can see that, again, temperature and our concentration of magnesium, as well as our inorganic phosphate, are going to play a role. All right. So if we look at concentrations that are in the cell of our various adenine compounds, ATP is the highest, typically. ADP is kind of in the middle, and AMP is low. So if we convert ATP to AMP, suppose I take 0.1 ATP concentration, convert that to AMP, This is going to change a very small amount, like 3%. So that's not a big change. That's not going to be noticeable. But here we'll go from 0.29 to 0.39, which is about a 33% increase. So small change in ATP results in a big change in AMP. So we could look at changes in ATP as a measure of the energy balance of the cell, but a lot of times we'll look at AMP because we'll see bigger percentage changes in there. So a 3% change, we might not be able to detect that very well, but a 30% change, that's going to show up really evenly. So AMP is often used as a sensor of the energy balance of the cell. So if our AMP levels start getting too high, we're going to see a lot of regulation kick in, assuming no disease states. Okay. Also look at the phosphocreatine in here as well too. But, anyway. The other thing to note about this is our ATP concentration is usually very far above equilibrium concentration. So ATP is a very good source of chemical energy for the cells. Okay. We actually will see that our phosphate groups, the hydrolysis of phosphate groups, tend to produce a lot of energy. So we'll see more than just our nucleotide triphosphates. We'll see other phosphorylated compounds that make high energy compounds in there as well too. For example, phosphoenolpyruvate, PEP, it produces a lot of energy, so it puts some PEP in your step or something in there, right? We'll see more of this when we talk about glycolysis. And if you look at this, the delta G for the hydrolysis of phosphoenolpyruvate is about double that of ATP. Why don't we use more PEP? Well, it's just that ATP is sufficient in high quantities, and PEP, ATP does a lot of stuff. PEP, we'll see where that comes in a couple places in there. Same thing like phosphocreatine, phosphoenolpyruvate, 1,3-bisphosphoglycerate. These are all somewhat higher energy than ATP. And the neat thing is we could actually burn those to make ATP if we needed it. But our ATP is also high energy. So we can get down here and, when we get into the negative side here, use those to synthesize other compounds. So we can use phosphoenolpyruvate plus ADP, take that phosphate group off there and add it to the ADP to make ATP, right? So this is just kind of a hierarchy of energy. We'll probably talk mainly about ATP being used to do this, but again, phosphocreatine, all that good stuff in there. Okay. So, a lot of times we write things as a one-step reaction. Okay, here we have glutamate, and we're going to convert that to glutamine. We're just basically going to add this amino group to it, and oh, look, we got glutamine. So we write that for convenience as a one-step reaction, but what really happens is, we require ATP for this, but the first step is we're going to phosphorylate this oxygen at the on the side chain of glutamate to give us this, glutamyl phosphate in here. All right, and then glutamyl phosphate is a good that makes the oxygen much better to be attacked by the amino group in there. So and phosphate is a good leaving group. So we get inorganic phosphate that leaves, and now we get our glutamine in here. Okay. So this is just to keep in mind, we have these pathways. Sometimes they're very long pathways. A lot of times we'll write, A goes to B, goes to C. Then there's 6 reactions, and we get to D, and so we will often kind of cut out some of this intermediate stuff in here for clarity. Okay, so. But if we wanted to be really, really super specific, we'd say, well, the first thing that has to happen is we need to phosphorylate this oxygen here, and then our ammonia can attack the phosphorylated oxygen and kick off pyrophosphate to give us our glutamine. So that's just kind of convention in there. If we look at thioesters, so we will talk a lot about things like acetyl-CoA. So we're going to have this sulfur group in here with the coenzyme A in there. We can hydrolyze this to get rid of the thioester. So we basically get CoAsh, coenzyme A that is thiolated in there. So we end up with acetic acid, and then of course we can isomerize this to give us acetate there. So the hydrolysis of acetyl-CoA, also very favorable, very similar to the ATP in there. Okay. The reason why this is important is we're going to see acetyl-CoA in a ton of metabolic pathways because acetyl-CoA is a great donor for acetyl groups. Here we have our nice little 2-carbon unit. So we want to add 2 carbons at a time. That's our acyl group. We can add that. When we see fatty acid synthesis, when we make fatty acids, we're going to add 2 carbons at a time. Okay? A lot of times when we look at fatty acid oxidation, we cleave 2 oxygens at a time. That works great if you have fatty acids that have even number of carbons. What happens if you have an odd number? At some point, you're going to have 3, and then we get some other crazy stuff that goes in there. Extra steps. All right, it's like overtime in games. It's like, hey, bonus hockey. All right, our redox reactions. Here we can see we have our alkanes. We can oxidize that to an alcohol. Then we can further oxidize that to an aldehyde or a ketone. We can further oxidize that to a carboxylic acid. And then finally, we've completely oxidized the carbon to carbon dioxide. Okay. So, and of course, we're transferring electrons in here as well, too. Need a second here. I don't know if it's the weather, all these different fronts moving in and whatnot, but yeah, I was just sitting on my computer working last night, and all of a sudden I was like, boom. Oh, okay, so still feeling funky today. Yeah. Like I got a pouch full of drugs in here, so I'm gonna have my ibuprofen, which I refer to as vitamin I. But yeah. We can also reversibly oxidize alcohols to ketones. Okay, so if we take lactate, we start working out and go out, shovel some snow or pry up some ice. I was gonna say, at least my kids had fun yesterday. I got them to clean off the front porch and the steps because they were seeing how big of a chunk of ice they could pry up there. I got a little crowbar and showed them how to do that. So, but you do that, you get lactic acid, and you can oxidize this to make pyruvate. And of course, this is reversible, and we'll see this, lactic acid to pyruvate, pyruvate back to lactic acid, and all that good stuff in there. So if we look at glycolysis in the muscles, you end up with pyruvate, which gets converted to lactic acid. That lactic acid will go to the liver, where it can get converted back into pyruvate and be used to make more glucose to send back to the muscles. So we're going to see compartmentalization as well this semester here. And then, of course, we'll often see stepwise transfers of protons and hydrides. All right, so good stuff. We won't talk too much about reduction potential, but just like we saw with our delta G for free energy, our reduction potential will also depend on the equilibrium constant in here. So we can actually relate our delta G to our electric potential in here. So delta G is essentially our molar constraint times a constant times the change in electron potential. What was really fun is, as a grad student, I actually titrated electric potential versus position or conformation. I had different mutations in there where I'd measure the various electric potentials of them and try to get a hyperbolic curve. It was pretty crazy too, because like the first paper I ever wrote, we had to resubmit like 2 or 3 times to get it published because apparently it was controversial. But when I did this electric potential titration, I could not get a true sigmoidal curve. I just had a hyperbolic curve because I couldn't get the electron potential low enough for the other side of the curve for one of the 3 curves I had. So I had this big explanation about it. I was like, yeah, it's like, oh, it's like theoretically this trend should continue. I just can't physically do this. I was waiting for the reviewers to go, well, we got a problem with this figure. No, that went right through. I was like, what in the hell? Like the paper that has really strong evidence, that one got hammered by reviewers, and the one that I thought was weaker went right through. So, oh well. All right. So standard reduction potentials are important for a lot of biological half reactions When we talk about ubiquinone making ubiquinol and semiquinols in here, I love this. We have 2 electrons. Sometimes we're going to see just 1, but we're not going to sweat these electric potential values. Just want you to be aware that they exist. So I talked about ATP. We also have NAD and NADP, which are very important redox cofactors as well too. These are our pyridine nucleotides. They can dissociate from the enzyme, so they are often cofactors on enzymes and will dissociate and can be replaced. So we can often transfer a hydride. So NAD plus, if you add that H minus to it, becomes NADH, right? And of course, this is reversible. Same thing with NADP plus to NADPH. Okay. All right, so these are kind of like what they look like. And then we can see our addition of protons or whatnot in here. Okay. Pardon me. All right. If we look at NADH, we can monitor this spectra photometrically. If we look at oxidized NADH versus reduced NADH, we don't want to measure this at 260 because it's not a very big change, but if we measure this at 340, our reduced one has a peak. And if that disappears, then we know we have NAD plus in there. So we can look at the changes in concentration at specific wavelengths. All right. Ooh, quantum chemistry. You guys ready for some quantum dynamics? All right. Very brief in here. A lot of times we'll see things in singlet spin states, but oxygen has that triplet spin state. When I do my lecture for the first semester of chemistry, I talk about water as being the most magical special compound because it expands when it freezes and all that stuff in there too. Well, oxygen is close behind that because this triplet spin state results in a lot of oddity in there. For example, you can't directly transfer an electron from a singlet reduced species to a triplet species. That's unfavorable according to quantum mechanics. So we don't see this direct oxidation occurring. Again, spontaneous combustion, not going to happen because it's prohibited by quantum mechanics. But you can do some, a lot of cofactors will get around that. That's how we actually do that for our reactions in there too. But then there's also some weird quantum mechanics, that whole spooky action at a distance stuff from physics. So maybe we get some weird tunneling and poof, spontaneous combustion. All right, enough of my conspiracy theories. All right, one of my favorite compounds, flavin cofactors, also allow single electron transfers. Why is it one of my favorite compounds? Because I get to say it like this, flavins. But our flavin cofactors are tightly bound to proteins, and you can see with this conjugated ring structure that they can help stabilize a single electron, a radical in there. So we have all this conjugated double bonds that one single electron can kind of get passed around and protected. I was joking with some other faculty who was considering having a kid, but is like far away from family and all that. It's like when you build a community and you find people to help with childcare and whatnot in there. I made the joke that I didn't even have to bribe students to take care of my child, for me. But that's sort of what we have going on here. This whole conjugated system, you can kind of pass the single electron around so that no one area gets too much of it. And again, we can see our semiquinone here. We have a single electron, and then if we fully reduce it by adding another electron, we get our FADH2. So FAD, FADH plus, and FADH2. So we can see there. And that's chapter 13. Okay, so that's our nice little review in there. This is probably the shortest chapter in here. I think there are 2 more that are close to it that are like 45 or 50 slides, but they're way far away. So that's why I wasn't too concerned about this first section in here. But the big takeaway here is we couple energetically favorable reactions with energetically unfavorable reactions. And we introduce some common molecules that we're going to see over and over again that are involved as cofactors, whether it's FADH2, NADH, NADPH. When we talk about the pentose phosphate pathway, we'll see our NADPH in there and all that good stuff. Okay. The big thing is redox reactions play a huge role in our metabolic procedures as well too, because we will see that flow of electrons in there, which is why I've changed up the order that we do these chapters in. Questions about any of this so far? Right. Chapter 13, we're going to jump to chapter 19, and then back to 14, 15, 16, 17, 18. So really, chapter 13 through chapter 19 are all talking about, producing energy, all right, for the cell. So because this is a short chapter and because we've already missed one day today, I'm going to go ahead and jump into the next chapter if I can figure out. Come on. There we go. I miss my multiple screens here in person. Okay. So let's go ahead and jump to chapter 19. Now, really, 14 and 15, I believe, are the first part of our energy producing sections. 16, 17, and 18 are the second part, and 19 is our third part, our payoff in there. So where's my Zoom? I got to share the screen now. This one, there we go. Okay. Yeah. So we're going to talk about oxidative phosphorylation, or really, I kind of renamed this chapter the electron transport chain, because we're not going to go into a whole lot of stuff about oxphos, because we can start getting into plant biochemistry, and we're just going to skip plants, right? Because we just can't cover everything. I mean, I could cover everything, but why do I teach this class the way I do? For those of you who are going on to professional schools or grad schools, you're going to get all this biochemistry from Biochem 1 and Biochem 2 in one semester. A lot of med schools and farm schools and some other schools will use biochem as kind of like their weeder course. So you don't go into nearly as much depth, but you get a bunch of information as fast as you can. For example, when my wife was in pharmacy school, her first semester, they said if you get more than 2 weeks behind, just drop. And, take out loans for more in there too. But if you get that far behind, it is, and so it becomes a lesson in how to balance things. The other thing is they can sort of do that because you won't have, like 7 or 8 classes, or how many classes do you have? You typically only have like 3 or 4. All right. So as opposed to the learning goals in that chapter 13, where we talk about bioenergetics and this and coupling and something, here it looks like we're not too bad here. We only have 4 learning goals. We're going to look at the electron transport chain, talk about the capture of light and photosynthesis. We can draw a line through that. Building up the proton motive force. Yeah, we're going to talk about that. And the synthesis of ATP in mitochondria and chloroplast. Draw a line through that. They're very similar, but so we really only have, the electron transport chain in my mind encompasses these last 2 as well too. So that's why I call this the electron transport chain. So oxidative phosphorylation, what we're doing is in our first 4 components of the electron transport chain, we are building up this electromotive force, this proton motive gradient. We're going to pump protons out of the mitochondrial matrix into the inner membrane space. Recall that mitochondria has 2 membranes, so we put all these protons in there. Our first 4 complexes do that. Our last complex lets the protons back in, kind of like water over a dam, which drives the machinery that makes ATP. So we use our energy from reduced fuels to synthesize ATP in animals. And we do this into mitochondria. Mitochondria are the powerhouse or the batteries of the cell for pretty much all organisms, at least all eukaryotes. So if we look at things like carbohydrates, we can burn our sugar. Oh, I already have my Mountain Dew today. It's a later day here. So I got my sugar in there. I have our lipids. If I had a big plate of bacon, and proteins, amino acids in there, we can reduce those for energy in here. So the electrons from those reduced fuels get transferred to reduced cofactors like our NADH or FADH2. So for the oxidative phosphorylation, okay, so oxidative meaning we're sending our electrons to oxygen ultimately, and our phosphorylation, we're going to phosphorylate something, in this case, our adenine, to make our adenine triphosphate in here. So we get the energy of that electron transfer from NADH and FADH2 to make ATP. Okay, so everything we're talking about in this chapter, how do we make ATP? Sound good? Yes.
So last semester, I was using the recordings and to
yes.
Like I would usually go back and watch the recordings and those that study guide. And I like what you say.
Yep.
You go more in depth. So they really post this.
Absolutely. Yep. Yep. I'm recording now because I know I've got at least one student who's quarantining. So, I will post these, and I will try and remember to do this every time. But you might come in and remind me. But yeah. And that's the key to this class is to keeping up. This is absolutely a class where don't fall behind because it gets hairy. All right, we're going to try and make it better. Okay, so if we did this in the normal order, chapters 13, 14, 15, 16, 17, 18, well, we'd see here in 14 and 15, we talk about glycolysis. I think 16, 17, 18, we get into the citric acid cycle in there. And then boom, now we get to this point. So we're really starting with the third stage of energy production in here. We'll get to these, but I don't want to put them all together ahead because I think talking about bioenergetics, that connects very favorably with the electron transport chain because we're going to see, FADH to NADH and our ATP. We're going to see examples of this energetics in action. Okay. All right. But the big thing is, we eat stuff and we produce these electrons. These electrons, can then, we produce more through the citric acid cycle. These electrons are all going to go towards making our reducing coefficients here, NADH and FADH2. And so these are our reduced electron carriers. They go into the electron transport chain and they help drive this formation of this proton motive gradient, electrochemical gradient, so that we can make ATP and have our energy. It's like, how do we make cake or dessert? It's funny, I don't even really like cake, but yeah. Anyway, if we want to actually add a pyrophosphate to an ADP to make ATP, this is very thermodynamically unfavorable. We saw that cleavage of a phosphate group, that hydrolysis of a phosphate group, really energetically favorable. That comes off, it's like, over 30 kilojoules per mole of energy is released. So you got to put in a ton of energy to hook that on there. All right. So if you try to do that in one big step, it's hard. So how do we do that? We don't actually do a direct reaction. Now we could use like phosphoenolpyruvate or phosphocreatine and directly just force it on there because they are very high energy. And we'll see that in very small concentrations that happens. But then you get to the point of, well, how do I make phosphoenolpyruvate or phosphocreatine? So it's just kind of like passing the buck on down the road. ATP is going to be our energy currency of the cell. So we want to find I don't want to say an easy way, but a favorable way of making ATP. It might be kind of a convoluted pathway, but we're going to just incrementally get there. So it's kind of like, here we are on the 5th floor. We could have like little spikes on the outside. You could climb up those spikes and it takes so much energy, that'd be a pain in the butt. Or we have stairs. You can take one step, if you get tired, you can kind of rest on the landing or something like that. But, you have these steps that lead you to the 5th floor until you get here. Or you can take the elevator. Okay. But again, it's incremental steps to get there. So what we do is we build up, this kind of reservoir, much like water behind a dam. We're going to build up all these factors that will allow us to have energy to make them. So we're going to have this flow of protons down an electrochemical gradient that is going to drive kind of literally biochemical machinery to attach that phosphate to ADP to make our adenosine triphosphate. Okay. All right, so let's see how this works. Okay. So to get this chemiosmotic energy, again, remember our osmosis, our membranes around the cells are like semi-permeable membranes. So we can use them to separate things over here, and our pores can allow them to come in. If we think of our transport across the membrane, sometimes we just open something up and let it come in. A lot of times it's a little more regulated. You guys ever you can get like little gumballs out of the gumball machine when you were kids? Yeah, you guys can't do what I did as a kid. You can't kind of reach up there and manipulate it because they've changed the machinery. I'm going to tell you a terrible story about myself and my classmates in middle school. There were a big group of us that were up for some award for like student integrity and something like that. So we had to come in on a Saturday and do these interviews. And so they kept us in a lounge that the teachers had access to, and it had a Coca-Cola machine in there. And in those days, if you had skinny arms like me and my friend Teresa, yeah, you could reach up under there and pull out the Coca-Cola and get Coca-Cola for free. So, for our ethical award, the two of us were in there stealing Cokes for everybody. It's terrible. But, if you think of the gumball machine, the way they work now is you put your quarter in and you turn that little crank and it opens up a little hole and the gumball falls down in there. You keep turning the crank, it comes over here, and then it drops out. We're actually going to see this kind of cranking mechanism for the formation of ATP. It's literally, instead of gumballs, it's protons that are coming through this little pathway, driving this spinning mechanism that provides the energy to make ATP. All right. Whew. Okay. So in bacteria, they use their plasma membrane. Yay. Okay, we'll note that, but we're not going to look at that in any great detail. Chloroplasts, they have the thylakoid membrane. Okay, we don't care about plants. We're going to look at exclusively the mitochondria in here and just say these are very related, very similar across those. Okay. Our membrane separates our two quantities. So high concentration up here, low concentration down here. So if we do have an opening, our gradient is going to flow this way. Okay. So then we need a protein that's going to couple that downhill flow of electrons to the uphill flow of protons into that center, so that later on we can have the protons flow in and we can make our ATP. Okay. All right. So that's what we see here. All right. So in short, our mitochondria looks like this. We're going to have 4 complexes, which we're going to conveniently name complex 1, 2, 3, and 4, that are all involved in pumping protons out of the matrix into that intermembrane space. And then complex 5 lets the protons back in, but couples that flow in with the formation of ATP. These 4 complexes are making a chemiosmotic gradient in here, electrochemical gradient. Why is it an electrochemical gradient? First of all, we have a higher concentration of H's in that intermembrane space than we do in the matrix space. So they want to flow down from concentration. We also see that we're pumping protons, so they're positive charges, so that intermembrane space is going to be positively charged, and our matrix is going to be negatively charged. So we have an electrical component. We want the positive charges to flow down there as well too. So there's 2 factors that want those protons to flow back in: a chemical gradient and an electric potential. Both of those together give us an electrochemical gradient in there. So they really want to flow in. Okay. All right. You go back to like the Matrix movie, and they're using humans as batteries. You say, well, see, here we have electric flow. You could technically use people as batteries. It could work, man. I'm going to go off topic here, but anyway. So this summarizes everything that's going on. All right. So complex 1 looks like Florida. Complex 2, no, I don't know what. Complex 3 is just huge, and complex 4 is kind of a big. Looks like Utah. No, okay. All right. So if we go back into our mitochondria, I got to check on my time. I want to make sure I don't. Okay, we're good. We got about 10 more minutes here. We got that double membrane. Okay. So the outer membrane is relatively porous and lets a lot of stuff through. A lot of metabolites. It's easier to get through the mitochondrial outer membrane into the cytosol. All right. The inner membrane space has an environment that's very similar to the cytosol, but it's going to have a very low pH because you're pumping all these protons in there. So it's got a lot of H plus concentration, higher proton concentration. You're going to have a lower pH. It's going to be very acidic in the inner membrane space. The inner membrane is much more impermeable than the outer membrane. it's a lot harder to get across it. So it's, I'm going to be political, but not really. For example, the U.S.'s southern border, it's pretty easy to get across the southern border there. And, but if you go to Canada and try and get in there, and it's funny because I look at my wife before she was a U.S. citizen, spent like 2 hours at the Canadian border because they were like, let's check your papers and all that. So it was a lot harder to get across that border there too. So our inner membrane is a lot more challenging to get stuff across, which is why we have a lot more pores and electron protein complexes in there. Also, got to keep in mind that the inner membrane, we kind of draw these as big circles, but our inner membrane actually makes crista. So we have all these little sections in here. So our inner membrane has a lot of surface area. Our inner membrane is just packed with protein as well too. I joke, it's like Snickers bar, it's packed with peanuts, but something like 70% of the mitochondria is protein, because these are all proteins geared towards getting things in or out of the inner matrix in there. And the matrix space is where we make our ATP, but it's also the location of our citric acid cycle and parts of lipid and amino acid metabolism as well too. So we need to get other stuff in and out as well. This chapter, we're mainly just focusing on the electron transport chain, but when we go back, we're going to see, oh, we got to get the citric acid cycle. Part of it's in the cytosol and part of it's in the matrix. Why? That'll be a question I'll have for you guys. And also our matrix has a lower protein concentration, so it's going to have a higher pH than the inner membrane space. So this is what I mean by the crista. You have all these little things that kind of stick out in there so that you have more surface area, so you can have more of this stuff going on and on and on. And this is actually kind of neat. I'm kind of assuming from biology classes you've gotten a lot of this structure in here, so I'm not going to go over it in any huge detail or ask a lot of questions about it. But you often see our ribosomes in here as well too, because again, we're looking at amino acid and protein formation, metabolism going on in there. There are a lot of soluble metabolic intermediates in the matrix space. There are a lot of things that are membrane bound as well too. And we're going to see when we talk about cytochrome C in the inner membrane space, it's loosely associated with complex 3, but it can roll over into a niche in complex 4. And if bad things happen, it can be jettisoned and released to the cytosol, which is a signal for apoptosis to occur. And then these are just different stains of the mitochondria. You can see electron micrograph. If you ever look at in a microscope, mitochondria look very dark because they're just packed with protein. There's so much electrons stuff in there, like NADH and FADH. It's real electron dense, so they show up very, very dark in electron micrographs. We can stain for our crista. We can stain for our I believe this is microtubules around there. Pull this up. Actin, yes. So that yellow is the actin in there that is connected to the mitochondria. So this is a major depot on the way in there. Oh, I love this. Add table 19 too. Whoops. All right. So the electron transport chain has a series of electron carriers. All right. So complexes 1, 2, 3, and 4. We're all going to see electrons flowing through them. Each complex has multiple redox centers, which will have either a flavin mononucleotide or a flavin dinucleotide, so FNN or FAD. We'll have a variety of cytochromes in there. There are cytochromes A, B, and C, and then sometimes there's B1 and B2 or A1 and A3 or something like that. We'll have iron sulfur clusters. All of these are redox centers, and they're geared towards shuttling along electrons, okay? Because it's the flow of electrons that's going to drive the creation of this electrochemical gradient, okay? And we want to make the potential between these areas just enough so that the electrons will go here. And it's like, oh, I want to go here. Oh, I want to go here. Oh, I want to go here. It's kind of like the psychology of how do they get you into a store to buy a certain thing, entice you into the store opening. It's like, oh, well, here's this. You look at, or I tend to think this more as like train depots. Okay, I'm going to go to this station, and then this station, and then this station, and this station. I probably should say subway stops, but first place I ever rode anything on rails was in Europe, so it was like train stations. All right, and then the order of transfer of electrons is dependent on the electron, on the reduction potential. So we're going to move just incrementally. Okay, So we can see stepwise by stepwise. This way, I don't have to take like one big step from the 5th floor down to the parking lot because that first step's a doozy. No, I can go down the stairs out there and just, stepwise to get down to the ground floor. Okay. Typically, we can try and donate 2 electrons at a time because that's nice and stable, but sometimes we got to donate 1 electron at a time to acceptors that can only handle 1. Okay. So we have like a great big elevator or a very small elevator, depends on how we load them. Our cytochromes have this protoporphyrin ring in them. Okay, so you can see if they're carrying electrons, we have this big conjugated ring structure. We can hold our electrons very, very well to get them transferred in there. Okay, if we look at cytochrome C, oxidized and reduced, we can kind of keep track of that in there based on the spectroscopic. If we look at iron sulfur clusters, so I studied a 2 iron 2 sulfur cluster in here in the Rieske iron sulfur protein. It was coordinated by some cysteines, or in my case, 2 cysteines and 2 histidines. But you have these things that will coordinate in here. You can have a single iron center, a 2 iron 2 sulfur center, or a 4 iron 4 sulfur center in there as well too. But again, these iron sulfur clusters serve the same purpose. That iron is a big honking electron dense area. So we can put an electron on there and then we can pass that electron along. Okay. We also see some mobile carriers in there as well too. Ubiquinone, which I will talk about way more than we need to, but it's a liquid soluble, so it can kind of like, a lipid soluble, sorry. So it can kind of move through the membrane on its own. It's not attached, but it's hydrophobic, so it'll slide through the membrane to transfer an electron from one complex to another complex. It readily accepts electrons. It's kind of neat. When it picks up 2 electrons, it'll also pick up 2 protons to convert that ubiquinone, so that that one becomes an ol to an alcohol. Okay, so it carries those electrons and those proteins from basically one side to the other. It's like a little shuttle bus. Electrons go this way, and they bring along the protons with them. So coenzyme Q is a mobile electron carrier that transports the electrons from complexes 1 and 2 to complex 3. Then it goes back and picks up more. It's more complex than that because it says it can take 2, but it only gets 1 at a time. So you make a semiquinone, which then has to go back up and pick up another passenger. It's like, the shuttle bus from an airport. We got to pick up a couple people. We got to make a couple stops before we get to our destination. So that's what we see here. In our first case, our ubiquinone, fully oxidized. You can see we have our ketones here. We pick up 1 oxygen and 1 proton. So now we have an alcohol group and a radical stabilized by our conjugated ring structure in here. We pick up a second electron and a second proton, and now we have a double alcohol, our ubiquinol, QH2, fully reduced, and we can go drop them off and regenerate our ubiquinone to go back and do the same thing again. Okay, so I think I will end with this slide, but let me talk about this slide. Our first 4 complexes are all involved in establishing that electrochemical gradient. We have complex 1, 2, 3, and 4, which is probably how I'll refer to them most of the time, but it's good to know the other names. Complex 1 is NADH dehydrogenase. All right, so it's going to be involving NADH, and it's got a dehydrogenase activity. So these all facilitate the formation of that electrochemical gradient. What you will see is complexes 1, 3, and 4 all pump protons across there. Complex 2, succinate dehydrogenase, carries electrons, transfers them ultimately to complex 3, but it doesn't pump protons. But it makes that ubiquinone into an ubiquinol, so indirectly gets them across there. Succinate dehydrogenase is also part of the citric acid cycle, so we'll see it again. So it's kind of got double duty, which is why it's not required to pump those protons directly across. Complex 3 is also known as ubiquinone cytochrome C oxidoreductase, or the BC1 complex. It's a mouthful, no wonder we call it complex 3, but it is a major, it basically 1 and 2 both separately feed into 3. So you can go 1, 3, 4 or 2, 3, 4. They both get electrons to complex 3, which pumps protons across the membrane. but it sends those electrons to cytochrome C, which isn't its own thing. It's kind of a loosely associated with complex 3. And then it receives those electrons from complex 3 and it rolls over and gives them to complex 4, our cytochrome oxidase, which again pumps protons across as well as letting these electrons flow through. And oxidase being the last one in there is where we're going to see our electrons ultimately going to oxygen there. You'll see that these things have varying numbers of subunits in here. Like NADH dehydrogenase has, a ridiculous number of subunits. We also have a lot of prosthetic groups in here as well. So flavin mononucleotide, flavin dinucleotide, various hemes, iron sulfur centers, copper centers as well too. All these are playing a role in getting these electrons traveling through their various little steps to help pump the protons across the membrane. Seems like there's one other thing I wanted to say. Okay. This is a little bit of a lie because our complex 3 is really a dimer. So it's got 11 subunits, but there's 2 of them, so there's really 22 in humans. And it's neat because you get one subunit here, and then the iron sulfur protein kind of juts out this way into the other dimer. So they cross like this, and then those iron sulfur clusters move like this to transfer. They basically pick up an electron down here, transfer it up here, then the electron ultimately makes its way to cytochrome C. I could talk about that for months, but I won't, I promise. All right, so where we'll pick up on Tuesday, I think I can finish this chapter on Tuesday, and then we can have our first general discussion about this electron transport chain next Thursday. We'll pick up talking about these individual complexes. All right, so what slide are we on here? 17 to 63, so that's not too bad. Well, we did what, 50 some, so we got to do like 45 on Monday. So that won't be too bad. Or Monday, Tuesday. It's going to take me like a month to figure out, especially with, a week that has a holiday and then a bunch of snow days. I'm like, what day is it? What time is my class? So yeah, we'll get there. And I'm going to open up the homework for this chapter as well too, so that you can take a look at it. Again, none of the homework is going to be due until our first exam, which covers 4 chapters. We'll go more into that as we get closer to the exam. But, I can let you guys go. Oops.